Welcome to those who want to have fun with histology made easy with drawings and diagrams. Today we're going to talk about connective tissue. Connective tissue seems like some kind of a fibrous tissue here and there, or doesn't seem that very important by itself. Well, connective tissue is a tissue that helps other tissues function adequately. It fills and reinforces other tissues. Where is it? Where is it located? It's all the way from the head to your toes, everywhere. It is everywhere. And the cell that is the most significant, pretty much, in the connective tissue is the fibroblast. The fibroblast is the jack of all trades, like we would say. It's able to do so many things, including, the connective tissue through other cells, bone and cartilage, our skeleton, our cartilage. So we'll start with the fibroblast itself. The fibroblast has a peculiar shape. Let's make it like that. This would be the nucleus, and you would ask yourself, why this shape? Well, this shape is nothing in particular. Usually this cell is in between the fibers that it produces, so it gets to be kind of collapsed by the fibers. That's why it's mostly elliptical or spindle-shaped. But they may be more rounded or even stellate-shaped at other occasions. What does the fibroblast do? Let's just, before doing that, I'm going to just a little bit darken the area of the nucleus of the fibroblast. What does the fibroblast do? Essentially, the fibroblast does two things. It produces a ground substance that is like a gel-like material and a good number of fibers. With those things, depending on which is predominant, you will find the connective tissue all over the place. For example, if we kind of want to show the gel-like material, I don't have a means to make it different, but it is something like a gel, pretty clear on H&E, and it is formed by sugars. The ground substance is basically made of sugars. Were called glycosaminoglycans before they were called mucopolysaccharides. They're just repeating units of two sugars, uronic acid and N-acetylglucosamine or N-acetylgalactosamine. In other words, two sugars in chain forming polysaccharides. Now, perhaps the most common of these glycosaminoglycans, we just put it here, GAC, is the hyaluronic acid. Well, which is composed of glucuronic acid and N-acetylglucosamine. In other words, we're dealing with a gel made of sugars that is going to fill up spaces whenever it's needed, and whenever it's needed also will give room for fibers. The fibroblast produces fibers, which all of them are proteins, up to an astonishing number of about 27 of them. Thank God we don't need to memorize all of them, only about a few of them. And how are they produced? And which are these fibers? You probably have heard about it so many times. And we'll start with collagen number 1, which is the most abundant. I'll put it over here, number 1, which is the one that's also present in the bones, and it is made of, it's eosinophilic. In other words, with H&E, these fibers are very thick, and they are going to appear as very, as fibers, as very thick fibers, or thin, depending on the tissue, that will give support to the tissues. Let's, for example, also, let's kind of go, how are these fibers made that are so big as to be present in some tendons, like in the biceps tendons, or the patellar tendon, or the Achilles tendon, that are so strong and made of those fibers, including the capsules of the joints and the ligaments that join bones together. Those are the functions of the fibroblast. How can a little tiny cell, like a mosquito, produce such a column of protein that is so extraordinarily strong? Well, if it's a protein, you know how it's produced. The recipe for the protein is in the DNA, obviously, which is by the messenger RNA. Is being copied that message to the rough endoplasmic reticulum, like any other protein that you know, which has many ribosomes, and then you start having that 3 polypeptides are being produced, 3 alpha polypeptides, that are going to be transported via vesicles to the Golgi apparatus. During that process, those polypeptides are going to be transformed, modified, changed a little bit, and finally it will be secreted as 3 chains. And let's try to kind of visualize 3 chains here in different colors. One of them will be like this. Let's put red for the other one. And it will be like this, for example. And let's kind of a blue for another one. So we get actually 3 polypeptide chains. And at the very end of that procollagen, we have some peptides at the very end here and here that need to be removed by a procollagen peptidases, that are enzymes around the cell, so that that molecule will be free to polymerize. Once the peptides are removed, this molecule will be able to polymerize, to join other molecules head to head and laterally to form those strong ligaments that you know in many parts of the body. The collagen is usually, like I mentioned before, is usually eosinophilic because in general, most proteins in the body on H&E appear red, and the collagen appears like that. For example, like that, you can see it in the skin, as in the reticular dermis, as crisscrossing bundles of collagen. And the other places, for example, that the collagen is less abundant, and I will mention one place, for example, which are the glands of the endometrium of the uterus, that are tubular type of glands, and the stroma in between, which is formed by fibroblasts, doesn't look like that thick. It's just it looks like a cellular stromal cells that give support, but it doesn't seem too very important. But those glands wouldn't be there if the fibroblasts wouldn't support them, and the stroma. Because with the stroma, you have vessels, you have other inflammatory cells, and also nutrients can diffuse. We are actually, you can perceive that this gel-like material doesn't at all limit the presence of vessels and other cells. Let's make, for example, a drawing of a vessel. You may have a capillary that is cruising through the collagen and is bringing red blood cells, white blood cells, oxygen, etc., which is going to diffuse to all these tissues. And you may also want to have a group of epithelial cells adjacent, because you have to remember epithelial cells have no vessels. They are just devoid of vessels. In order to draw epithelial cells, you can do something like this, like draw a basement membrane which actually has 3 layers. We'll just mention the fact that they have 3 layers, and obviously a set of other epithelial cells that we can draw on the other side, so we can actually remember that the basement membrane is there. And for example, this would be epithelial cells that have no vessels but are separated from the connective tissue by the basement membrane. The basement membrane is made up by the fibroblasts and also by the epithelial cells, and we will talk about that a bit later. So far we mentioned collagen 1. If we go a little bit in that order, we would have to mention collagen type 2. And type 2, we'll have to set it apart because it is present in hyaline cartilage. The hyaline cartilage that you have in your nose, in the trachea, and many other places is made of this kind of cartilage. There are other types of cartilage that don't have collagen type 2, but the most important in hyaline cartilage is collagen type 2. What about collagen type 3? Also produced by the fibroblasts or other cells similar, is the collagen type 3 would be, for example, let's mention like that. Collagen type 3 are the reticular cells, reticulin fibers. And if you mention, for example, a few cells, if you have, for example, a few cells of the liver, of the spleen, of the lymph nodes, the reticulin fibers are going to be there whenever the tissues are very, very cellular, and we don't need to have big fibers. So we just want to have a framework of fibers. So we have the reticulin fibers, which are going to be difficult to see on H&E, but they can be stained with silver salts for reticulin. These are, for example, quite important in the liver, as we will see later. Now, we have also another type of fiber, which is the elastic fiber. There are some tissues that need to have this feature of being retractile, being elastic, and there are not too many in the body. And the elastic fibers, I will draw them with this color. Although they are also eosinophilic, I will draw them here. I'll have to erase the endometrium here. The elastic fibers are pretty much like an amorphous substance, almost like bubble gum, but that has the tendency to shrink. And obviously this is the elastic fibers. And they are present quite significantly in the big vessels like the aorta, on organs like the lung. Also in places like the skin that needs to retract. It has this elasticity. Elastic fibers that also are eosinophilic, but they can be stained by silver salts as well as the reticulin fibers. Now, since we are kind of having this situation going on, there are also fibers that are associated with the basement membrane, and this is the collagen number 4, and also collagen number 7. They are associated with the basement membrane. So the basement membrane is almost like a mattress where all the cells attach and get attached very firmly. And they include a number of substances like a hypoglycosaminoglycan, heparan sulfate, and also other little proteins like fibronectin and laminin and others. But anyway, when you look at the basement membrane on the electron microscope, you will see that it is separated, has a dense area in the center, lamina densa. They have a lamina lucida and lamina fibroreticularis. The inner portion of the cell of the basement membrane is tightly attached to the tissues. Well, after that, we could mention a few things. For example, like the connective tissue is a host of a number of things, cells. We say that vessels are quite abundant in connective tissue, depending on the place. Well, we have also other cells, like for example, let's mention here that are the histiocytes or macrophages. Are kind of rather large cells that their function is to phagocytize and be part of the defense of the organism, and they are believed to come from the blood monocyte. So the monocyte enters into the tissues and remains there. Also, you may have, for example, another types of cells, and we mention here, for example, like the white cells, like neutrophils, that may be passing by, and they are also part of the defense of the organism. We can also have cells like are kind of unusual, but they are present. You have to look at them carefully, and they are pretty blue. They have dense blue granules, and they're called the mast cells. The mast cells produce, they were thought to be the same basophil from the blood that stays in the tissues, but apparently they are a different kind of cell, and they are more, we don't really know exactly how they get there, but they are present in many tissues. They participate in certain pathologic conditions such as asthma or hay fever or even anaphylactic reactions. So the mast cells usually are not clearly seen unless you use special stains like toluene blue or some immunoperoxidase like CD117, which is good for mast cells. You don't need to memorize that. Other cells that are peculiar, but in specific places, for example, like in the lymph nodes in the outer portion, are cells like that have this peculiar appearance. More or less look like a star, and they're the reticular cells. The reticular cells are said to be part of the reticuloendothelial system in the body, and they may have phagocytic function or antigen presenting functions. They are cells that I'll write it down for you. The reticulum cell. Well. We are getting close to the end of this chapter, but we have to also take into account that some cells are also associated with connective tissue, although they are not really essentially connective tissue. And I will mention them here. I will draw them here, and are the adipose cells. I'll just make them here. The adipose cells, the white adipose cells, are the cells of our body that are store fat, and they are filled with a single vacuole without membrane, pushing the nucleus outwardly. When you look at your microscope, you'll see that those cells are totally clear, but that simply because the fat has disappeared during the processing. So those holes appear there. And they're important at storing energy in the body and giving the shape of the body. Being a little bit more abundant in women in strategic places, obviously. But this is the white adipose tissue that is present in us, essentially in different amounts. We have another type of tissue that we'll have to mention it here, that the cells are smaller. It's an adipose tissue, called brown adipose tissue. This is the white, this will be the brown. The brown adipose tissue has the nucleus more or less central or paracentral, and has this, when you look at it, a very brown color. The color is given by the great amount of mitochondria. The mitochondria and the fat globules, so they are kind of filled with mitochondria and little vacuoles of fat. What do they produce? They produce heat. And they are quite abundant in animals like the bear and hibernating animals that get to a long siesta during the winter, and they depend on this kind of tissue to give them some heat to go through the winter. Now, some newborns also have certain amount of brown fat, perhaps to protect them when they are born, to give them some protection from the cold. But that tissue disappears in a mature person, maybe you may occasionally find some clusters around the adrenal gland or maybe in the retroperitoneum, but they are not that abundant at all. In general, I would have to tell you that those cells that have abundance of mitochondria, grossly, they tend to look brown. So this is the brown fat. With this, I would have to end my little talk, and I advise you guys to go back to your books and enjoy the lecture. And now that you're going to understand most of it, and I wish you a great 4th of July. Take care.